Hello, biology scholars, and welcome to the first upload of the 2022-2023 school year. We are going to be talking about tonicity, which is the focus of the upcoming lab. For now, though, we need to start by figuring out exactly what tonicity is. So we're going to start by defining it. Tonicity is the ability of a solution to cause, in this case cells, to gain or lose a fluid, which in this case is going to be water. So when we think about tonicity in the context of cells, we're going to need to remember the process by which water crosses the plasma membrane or the cell membrane, and that is called osmosis. So when we draw membranes and we draw water moving across them, typically, if we were doing a more detailed sketch, we would have protein channels called aquaporins to facilitate the transfer of water. In this video, I'm going to simplify the sketches, meaning I'm not going to show you the aquaporins. And when we're talking about any solute concentration across a membrane, we always have to think about the concentration gradient. And if you remember, for our discussion about osmosis, water is going to move down its concentration gradient. It moves from areas of high concentration to areas of low concentration. What can be confusing is that this also means something else in terms of solute. Water is going to be moving from areas with low solute concentration to areas of high solute concentration. If you need a way to visualize this, we can make a quick sketch. I'm going to draw in a simplified membrane. And here I'm going to pretend that the solute is not able to cross over the membrane. It's stuck where it is. So, if I have a mixture or a solution on the left side that is 60% water and 40% solute, and then on the right side, my solute concentration is much higher at 60%, what that means is there's a lot less water. So instead of a 60% water, only 40% of that solution is going to be water. Now this is a very exaggerated case, but here you see that water is going to move down its concentration gradient. So I'm going to highlight that in blue to show the movement of water. And we're pretending that the solutes do not travel across the membrane, and they're stuck where they are. So what's going to happen is both of these sides are going to be able to reach an equilibrium when the solute concentration is the same. But to do this, we have to put water through the membrane to dilute the solute. There are 3 vocabulary terms that we can use when we are describing tonicity. We can say that a cell is being put in a hypertonic solution. Alternatively, we can put the cell in an isotonic solution. Or we can put the cell in what is called a hypotonic solution. Keep in mind that number 1 and number 3 are 1 letter off from each other. So in number 1, I have hypertonic with an E, and then in number 3, I have hypotonic with an O. That distinction is going to be very important, and it will give us a great method of how we can remember these 2 vocab words. In each of these examples, I'm going to have a beaker. And this beaker is going to be filled with some solution. For this example, I'm going to say that this is water with salt in it. So it's a salt solution. or NaCl solution. And in each of these, I'm going to draw a cell. I'll draw it a little bit rectangular in shape because when we work on our tonicity lab, we are going to be working with plant cells. So just as a reminder, this green shape here is representing a cell. Clearly, this image is not drawn to scale. Now we're going to add solute in, and we're going to start with an isotonic solution in the middle because that's the easiest one to explain. Let's say that in our isotonic solution, on the outside of the cell, we have 3 solute particles, which I'm representing by little dots. For an isotonic solution, the solute concentration on the outside of the cell is equivalent or equal to the solute concentration on the inside of the cell. So I'm going to draw in 3 solutes on the inside. So I have 3 on the outside and 3 on the inside. As a result, water is able to move inside of the cell, but this is only the case if we also have water that's moving outside the cell at the same time. There is no overall movement of water inside or outside of the cell. You can have water coming inside at the same time water leaves, because then you still have the same amount of water that you started with. So we'll summarize this in words. With an isotonic solution, the concentration of solute stays in equilibrium. And the same amount of water enters and leaves the cell, and this occurs at the same time or simultaneously. So really, you're not going to see a change here. And the cell's going to be pretty much the same shape. Let's switch over to the hypertonic solution here on the left. In this case, we're going to have a higher solute concentration outside the cell. So I made a lot of little solute dots there. But inside the cell, I have very little solute. So a much greater relative concentration of water here. But water wants to go down its concentration gradient. And if the solutes can't move, water is the only thing that can. So you'll see water exiting the cell. So to summarize this, in a hypertonic solution, the concentration of solute is higher outside of the cell. What this means is that the concentration of water is lower on the outside. So water moves down its concentration gradient, and we say that water moves out of the cell. If we wait long enough and enough water is pulled out, our cell will start to look kind of shriveled up. So instead of being a nice plump square, it's a little bit of a hot mess. Having the E in hypertonic, the E looks a little bit shriveled compared to the O of hypotonic. So I like to remember that the hypertonic solution shrivels the cells because of that shape that the letter E makes. So hypotonic is the opposite of hypertonic. So when we have a hypotonic solution on the outside, remember the outside is the solution, we only have a couple of solute molecules. But in this example, we have a lot of solute inside the cell. In this case, water is going to come into this cell. So the concentration of solute in this case is lower outside the cell. Or if you really want to think about this in the context of water, we can say that the concentration of water on the outside is higher. And what will happen here is you'll have water that will come into the cell. and this water is moving down its concentration gradient. If you have enough water that's rushing in to this example cell, it will go from a normal rectangular shape to a very swollen cell. If too much water comes in and the cell doesn't have a way to get it out, then you could see cells start to burst. That's why osmoregulation, or the regulation of water levels inside of living organisms, is so important. As an extra example and application for humans, we see that we can have blood cells, red blood cells, in hypertonic, isotonic, and hypotonic solution. Ideally, our blood cells are in an isotonic solution, meaning that an equal amount of water comes in as goes out of the red blood cell. This gives them their characteristic shape that allows them to carry oxygen to our cells. However, in some cases, if you put red blood cells in a hypertonic solution, you will get your red blood cells shriveling up like this. Or if we put them experimentally in a hypotonic solution, meaning that the blood cells have more solute inside than the solution itself, water will rush in and in some cases burst the cells open.